Okay, thanks for tuning in to this audio abstract. Today we have Martino Franchi discussing architectural, functional and molecular responses to concentric and eccentric loading in human skeletal muscles. So thanks for tuning in and thank you Martino.
Thank you, Rob. Thank you for having me on this series of audio abstract, which I find very, very nice and very interesting. I'm sure that people will start liking the idea more and more about this.
Good.
Yes.
Do you want to take us through the paper?
Absolutely, Rob.
Cool.
Basically, this is a part of my now PhD stuff, and it's one of the most interesting study I think of my PhD, wherein we investigated the architectural, the functional, and molecular responses of human skeletal muscle, in this case vastus lateralis. So by using leg press modified ad hoc, which I will explain to you soon, which I'll show you soon. Basically, the response to concentric or eccentric resistance training. What we found is that, the muscle growth with concentric and eccentric resistance training was then after 10 weeks of training was similar, but it occurred with different morphological adaptations and so architectural adaptation, which was reflecting distinct fiber fascicle behavior and molecular responses, which could potentially be linked to these different adaptations. Before starting everything, this is something that I wanted to show. Let me get slide up. So I wanted to show this slide because I wanted to explain to the listeners basically why we decided to adapt such design. So we decided to match concentric and eccentric resistance training for the relatively maximum load. And the reason why is because, according to the force-velocity relationship, every force-velocity curve, every point, every value of velocity and force should belong to the same curve. But this is potentially not met, this requirement, in conventional resistance exercise. So when we basically go to the gym and we go potentially on the leg press and we decide to do, push and so lift and lower a load. So what happens is that we go to the gym and we decide to work at 70% of the repetition maximum as an intensity. But usually what is a repetition maximum is the maximum load usually that we can lift. So it's kind of a measure of our concentric phase, of the maximum load that our concentric phase can bear. But if you see the curve, for example, in this case, we're on the 75% of neural activation force velocity curve. If we displace the same load between concentric and eccentric phase, knowing that during eccentric phase we can produce greater amount of force, we clearly fall into a different force velocity curve and potentially with lower neural activation. So in that case, we kind of have to de-recruit motor units to lower the load. And as just a kind of an example, you can see this is the rectified EMG of vastus lateralis during a concentric and an eccentric phase. And you can see that obviously the value of EMG, rectified electromyography, is bigger here in a concentric phase compared to the eccentric phase when we displace the same load. So that's why we decided to to use a greater load during the eccentric phase to compare the two resistance training groups and to match them, for relatively maximum load. So I prepared also a study design slide. What we did basically, we used this leg press. we use a unilateral eccentric versus concentric training design. We took maximum voluntary contraction, magnetic resonance imaging, and ultrasound scans before the start of the training and after the start of the training. We trained the participants for 10 weeks, 3 times a week, and we also assessed, and this is something I will talk later, the acute molecular responses 30 minutes after the first bout of either concentric or eccentric training with microbiopsy technique. Yes, so we train it with 4 series of 8 to 10 reps at the 80% of either concentric, as I explained before, or eccentric repetition maximum 3 times a week for 10 weeks. So this is quite good. We obtain ultrasound scans, we obtain a magnetic resonance scan, so we had a muscle volume as well, and so I would say that it would be quite nice to go through the results now, the paper. Or before doing that, so everybody can understand the design. So we had a leg press, which is modified ad hoc. You can see there's an electric winch which is attached to the leg press via steel cable, and that would allow us to allow the participant to do either a concentric repetition. So we were actually following them while they were pushing out, and they will stop in the chair without making him go back, basically the chair. Or we could have done simply the opposite. So taking them back with the chair without any sort of muscle action developed by them, and then we would have released the chair in order for them to make just a pure eccentric phase, pure eccentric contraction. So what happens after 10 weeks of training, either concentric and eccentric training? What happens is that we found the similar increase in vastus lateralis muscle volume, calculated via MRI scans. So eccentric training presented a 6% increase, and concentric training presented an 8% increase, this was not significant between the two groups, the two resistance training groups, and you can see that. As it wasn't different, the strength increased between the two groups, as the MVC increased very, very similarly between after eccentric and compared to concentric resistance training. But what was striking to us is something that has been shown before by Reeves and colleagues in 2009 on older people, though. The architectural responses to eccentric and concentric exercise were almost diametrically opposite. So it looks like that eccentric exercise favors the increase in fascicle length after 10 weeks, and a concentric exercise favors instead the increase in pronation angle. And we know that architectural features play an important role in the function of skeletal muscle. This was very peculiar, because basically you can see they reach the same level of hypertrophy, but they reached in two opposite ways, in two completely different ways. Also, this at a cellular or sarcomere level, the increase in fascicle length likely reflects an addition of sarcomeres in series rather than in parallel, whereas the increase in pronation angle has been seen as a strategy to pack more contractile material in a given space along the deep tendon aponeurosis of a muscle. So likely reflects an increase in an addition in sarcomeres in parallel rather than in series. So this was the first bit that we found very interesting. As you can understand, if you're in a performance way, or even in a clinical settings, if you know that two different contractions have two different distinct architectural responses, then you can work out what you could need for that muscle in that specific moment, and potentially try to see if applying different types of contraction, you can have different responses and different functional output as well. As we know that, for example, if you put more sarcomeres in series, that should influence the velocity of contraction of a muscle. Another quite interesting result that we found was the vastus lateralis regional hypertrophy. So what we found is that when I went to analyze a series of like consecutive cross-sectional areas at different parts of the muscle, which I called in this case proximal, mid portion, and distal part. So I if I remember correctly, was 20%, 50%, and 70% of the vastus lateralis length around that. What I've done a mean of 5 cross-sectional areas per region, and I just checked if the increase of cross-sectional area was different between 2 different groups. And what I found, yes, it was the 2 different contraction brings 2 different regional hypertrophic responses, which I think is quite interesting as well on a well, functional point of view, of course, on a performance point of view. If you need to have like different parts of muscle which are going to be more interested in a specific movement, and this likely seemed to reflect also the architectural adaptation that we found that we found with ultrasound scans. Need to mention that one limitation of this study is that we have a snapshot of ultrasound just in the middle of the vastus lateralis. Although we use a 10 centimeter probe, so a rather long probe, so we could track at least 10 centimeter of fascicles and of vastus lateralis. Well, it looks like that because of the regional hypertrophy that is occurring, we should potentially have a look for different regional architectural adaptation, maybe in different sides of the muscle. Nonetheless, rather interesting result, I would say. What we wanted to also assess, so it was a little bit at the start. It was a little bit striking that the 2 exercises reached the same level of hypertrophy. Also, counting the fact that eccentric resistance group was using a bigger, load than concentric one. But this is not the first time that the same similar hypertrophy can occur at a result of an eccentric or concentric training compared to each other. But what we wanted to understand was, why do they need to what is the cause of why they need these 2 exercises bring to the same level of hypertrophy, but with 2 different pathways of hypertrophy, of architectural remodeling. So we checked if actually there is a shortening or a lengthening with the 2 phases, and that is well represented by this graph. You can clearly see that when the vastus lateralis does performs a concentric action, there is actually a shortening of fascicles, and the fascicles are actually instead lengthened during an eccentric phase. And this is something nice, which I always pardon me, but I always like to show this. This is quite clear in this ultrasound video. You can see that the fascicles are shortened and then they tilt in pennation angle, whereas during the eccentric action, the vastus lateralis fascicles are, lengthened, and you can clearly see that there's a lengthening of the fascicles. So there's 2 mechanical stimuli that are applied onto the muscle to clear different mechanical stimuli. So we thought, similar hypertrophy, different architectural adaptations, different mechanical stimuli applying to the muscle. what lies behind all of this. And that's where we went. That's why we went on the biopsy, the biopsy quest. Let's say we decided to measure the acute responses after 30 minutes, and we decided to look specifically at some proteins of the MAP kinase family. The reason why is because there were like similar, previous paper on animals that showed that the MAP kinase family is phosphorylated differently between these two contractions, concentric and eccentric contractions, and usually eccentric results in a higher phosphorylation of some of those MAP kinase of the family of the MAP kinase. Moreover, a paper on this is very interesting in my opinion. A paper on cardiomyocytes showed that when there is an overexpression, or when there is not an expression of this ERK1/2 kinase, basically the cardiomyocytes do respond by either like placing new material in series or in parallel, and this is very similar to what happened to what we think has happened to the skeletal muscle. So we thought about like looking at this target, and the results were striking. I found P38 MAP kinase to be 20, if I remember, like it was 23 fold more phosphorylated after 30 minutes of eccentric exercise, and you can see concentric there is almost non-response, as other proteins of the family were involved. So basically, as a conclusion of the study, we could conclude that let me get this open. So it was a similar increase in vastus lateralis volume, so similar morphological response, similar increase in strength, but with complete different remodeling patterns. And well, we found the MAP kinase family to be more phosphorylated and more activated after eccentric exercise and non-activated after concentric exercise. So we cannot basically say that this is the real reason. After this paper, I've been questioning if it was muscle protein synthesis differences, also differently along the muscle, and this is another publication in which I talk about this. We didn't find any differences, as papers before didn't find any difference in muscle protein synthesis. So is it a case that literally these two contractions have different, programmed response in muscle architecture? Could be the case, as my friend and colleague Ryan Timmins with his group has found very similar architectural responses in the biceps femoris, long head, so in another muscle group. So far we don't know, but I'm very keen to find out why this is the case. And as a practical application, Ryan Timmins talks very much about like injury prevention, and I absolutely agree, especially in a muscle that's got it always works in shortening, like the hamstrings. But even in the vastus lateralis, there are a lot of like injuries that occur, like also in the quadriceps. And practical application is that if really the muscle can respond to two different contractions in two different manners, well, it's good to know, especially for clinical, obviously, settings as well, but performance, because with muscle architecture we can influence the functional output of the muscle. So that's pretty much it.
That's cool, mate. That's brilliant. We've just run over a little bit, but that's absolutely fine. I'm just going to ask you to let us know where people can keep up to date with what you've got going on. I know we've communicated on Twitter,
Of course.
so you're on Twitter. Do you know what your Twitter
Yeah,
handle is?
my Twitter should be at mvfranchi, Franchi. I have
Cool.
I
I'll put a
usually
link.
use Twitter a lot to tweet papers. I really like Twitter as as a platform to share stuff.
Absolutely.
You can find me on ResearchGate. I have all the publication which you can read. I'm a believer of sharing of
Yeah, absolutely.
science. And well, my email is m.franchi@nottingham.ac.uk. And if you're interested in anything or you want to know more, you need like any PDF of papers, just contact me, because I'm very keen in this, on investigating muscle remodeling to different types of contraction. And at the moment we have like few studies going on aging wise, so that would be something I'm looking forward to publish, but for now I don't have the analysis ready.
No, that's cool, mate. That's absolutely fine. I just want to finally just thank you for tuning in and give us an overview of the paper, and we'll keep in touch and speak soon.
Fantastic, Rob. Thanks very much to you, and keep going with this audio abstract because it's very good.
Thank you very much, mate.
Bye bye
Speak
bye.
soon.
Ciao ciao.